I'd like to welcome Sir David Lane. He is the Chief Scientist at the Agency for Science, Technology and Research, Singapore. His talk is How the P53 Pathway Controls the Response to Chemotherapy. Thank you for joining us. You are credited as one of the scientists who discovered the P53 gene in 1979. Did you realize the importance of your discovery at the time?
No. I think I was very young and working in London and working on a tumour virus, SV40 virus, and it had a single protein that was responsible for its ability to transform cells in culture, to turn normal cells into cancer cells, which was called large T antigen. And we kind of went fishing with T antigen to find out what other proteins it would interact with, and that's how we discovered P53. I think we realized it was important because at the time we knew that this virus had to somehow specifically affect the host cell. But I couldn't have imagined that we'd be in the situation we are now with more than 50,000 papers on P53 and it being one of the most important genes in human cancer. So it's been an amazing story.
What role does regulating the P53 pathway play in cancer research?
Well, it's one of the principal tumour suppressors in man, so loss of P53 occurs in at least half of all human cancers, typically by mutation. In the remaining cancers, we find that the pathway that activates P53 is often lost, sometimes by epigenetic silencing, switching off of genes somatically. So it's a very critical tumour suppressor. It seems to work by protecting us from development of cancer because it can kill cells that have aberrant DNA. So I called it the guardian of the genome a few years ago because that's one of the things it does. If you irradiate cells, then they will die a P53-dependent death. And that role of protecting the body by eliminating inappropriate cells is clearly one of the major ways that P53 works.
Could you describe the results of your studies on how the P53 pathway controls its response to chemotherapy?
Yes, this has been a very important issue right at the start. As soon as we realized from work that we and others had done that P53 was a key part of the way that cells reacted to chemotherapy and to DNA damage, we hypothesized that tumors that had retained the wild-type P53 gene should be more responsive to chemotherapy than tumor cells that had lost the gene. And in preclinical studies in animal models, that seemed to be the case. But as we've turned to clinical trials, and a lot of clinical trials have now been done to say, should you segregate patients on the basis of their P53 status, the results have been confusing. So we've started to look at a lot more detail of what's going on, and it's complex, actually. The different chemotherapeutic drugs, which we tend to put in a single box as cytotoxic chemotherapeutics, tend to be much more subtle than we realized. So some of them activate P53, and that's very important for how they kill cells. And an example of a drug like that is actinomycin D. Whereas other drugs, like doxorubicin, that are widely used in the treatment of cancer, they do activate P53. But that's not the principal way that they kill cells. And paradoxically, as I'll discuss in tomorrow's talk, in fact, P53 can be protective against the effects of these drugs because it can cause the cells to stop dividing, and that makes them resistant. So what we're beginning to learn, which I think is very exciting, is that some of these so-called old drugs are much more specific than we thought, and that we might be able to use them in new and more sophisticated ways. And that's very promising. I think the other thing that's happening is this concept that we have of cyclotherapy. That is, we might be able to protect the body from the harmful effects of some drugs by placing cells out of the cell cycle. And if you imagine a tumor that's lacking P53 and a drug that activates P53 in normal cells, then we can have a situation where we can temporarily stop the normal cells in the body dividing. The cancer cells will still divide. They don't respond to this P53 activating drug and therefore retain their sensitivity to other drugs. And this is potentially very important. We might be able to protect people against hair loss, against mucositis, against bowel problems in their chemotherapy. So, it's a very different way of looking at our results and exploiting these genetic differences in cancer, but potentially very positive and exciting.
So how do these results impact the cancer research arena and potentially the selectivity of chemotherapy?
This is what we, of course, want, is to have chemotherapy be more selective, to be more appropriate for the patient. And there are still lots of mysteries. Why does the platinum drug work so well in testicular cancer but not so well in other cancers? Why have these chemotherapeutic regimes that are so successful in treating cancer have been derived really from arbitrary and repeated clinical investigation, but what's the molecular basis for those sensitivities and resistances that we see? So my hope is that by delving deeper into the pathways around the p53 system, that we'll be able to individualize even the use of existing therapies and radiation in a more selective way, so that we see a higher response rate and less side effects from those treatments.
Currently, a variety of p53-based vaccines have proven effective in animal models and are now undergoing human trials. What are your thoughts on the potential for immunotherapy in cancer care?
Well, I think immunotherapy is one of the things that's just undergone an amazing revival. I mean, just a few hours ago, we heard this fantastic talk about the use of antibodies to CTLA-4, which affect the T cells and the suppressor T cells, and they're sort of liberating a otherwise hidden and very effective curative response, immune response in some melanoma patients. So the whole field of tumor immunology is undergoing a huge change in confidence. And p53 is a very good target antigen because in many, many tumors the protein accumulates. It's normally present at very low levels in cells, but in tumor cells, as part of the effect of mutation and complex feedback networks, which I shouldn't explain now, but the protein increases in level and therefore becomes an attractive target. So I think the combination of tumor vaccines combined with the modulation of the immune response by antibodies such as the CTLA-4 antibody do offer a really exciting prospect. And we have to couple that with the fact that many of our current therapies are immunosuppressive, and as we develop more selective anti-cancer drugs, which are not immunosuppressive, then we may be able to find very virtuous combinations. I'm extremely excited about it. I started off as an immunologist, actually, and it was always the dream that we could use the immune system to tackle cancer. Great advantage of it is that it can hunt out the rare cell. So the exciting thing about these results is that we're seeing real cures rather than even prolonged delay of development of disease. So it's a very exciting time. And the whole field suddenly got a big revival, which is why science is fun.
Dr. Lane, thank you so much.
Thank you.